हेलो सर, यहाँ पे यूट्यूब दिखा रहा है स्ट्रीमिंग। सर वॉइस आ रही है? हेलो हेलो
बोल मेरे को थोड़ा सा ऑडियो मिल रहा है आपको? बोल रहे हैं अभी तो हमें दिख रहा है यहाँ पे जा रहा है।
हेलो हेलो माइक चेक। हेलो
आप बोलिए। बोल रहा है वो?
कौन? मैं तो बोल रहा हूँ, ऑडियो लगी हुई है।
आ रही है ना? हेलो बोलो करते से।
हेलो माइक चेक। हेलो
और उससे ठीक है, आप बोलिए। नहीं आ रही?
अभी आप जब बोल रहे थे तो आ रहा था।
एचडीएमआई कर दो ना।
इसमें
एचडीएमआई
HDMI तो लगा है, ना स्पीकर है। हेलो 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 चेक माइक चेक। हेलो हेलो माइक चेक। बोल रहे हैं।
बोल कर रहे हैं।
हेलो चेक।
बोल
हेलो
रहे हैं।
हेलो चेक।
वहाँ से तो म्यूट नहीं कर रखा है।
वहाँ से
यार वो टीवी हमें लगता है प्रॉब्लम टीवी है।
हाँ
डायरेक्ट
डायरेक्ट लगा क्या?
एक मिनट
अरे मैं कर रहा हूँ जहाँ से केबल में डालेंगे वो नहीं।
हेलो हेलो हेलो
आप
हेलो हेलो
हाँ
हेलो हेलो चेक चेक माइक चेक।
हेलो हेलो अशोक, मेरी आवाज आ रही है?
कोई ये लैपटॉप का डिस्टर्ब हो रहा था, इनवाइस कर दो उसको।
टीवी खराब है क्योंकि बीच में स्पॉट है। कोई कनेक्ट नहीं हो रहा है यार। दूसरा भी कनेक्ट हो रहा है तो अभी
तभी क्या है तो
किसकी?
हमने
नहीं मैं नहीं लाया।
वो करा सचिन
लाया नहीं। क्यों?
सिंह बोल रहा
हाँ?
था, सिंह बोल रहा था।
नहीं चाबी उसकी लाऊंगा, जरूरत नहीं थी। हेलो चेक चेक माइक चेक।
वही तो आवाज आई थी।
हाँ आई थी भाई आपकी आवाज। इसको भी हो रहा था।
आपको चलता है? ऊपर वाला में क्या कर रहे हो?
कैमरा सेटिंग कहाँ है?
हाँ?
इस कैमरे की सेटिंग कहाँ है? आती है ना?
नीचे कैमरा पे क्लिक करो।
बंद कर दिया। एक सेटिंग आती थी पहले।
इस पे जाओ वीडियो पे, नीचे तो कैमरा है ना
ये
zoom वाला।
चेक हेलो 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 चेक हेलो हेलो आवाज लग रही है?
मेरी आवाज नहीं आ रही है।
तो आपकी आवाज तो भी आ रही है ना?
उधर से
हाँ आ तो रही थी।
आई तो थी, वही तो कह रहा
यहीं से
हूँ।
गाना चला के देखो ना।
गाने चलाओ। हाँ गाने चलाओ।
मतलब किस लिए?
लोकल चला के देखो,
आवाज सही है,
मिलेगी कि नहीं।
आवाज सही है ना?
हाँ आवाज
यही यही लगा देना बस,
लोकल
बाकी
मशीन से आवाज भी मिले कि नहीं, वो देखो ना पहले। अरे कुछ भी चला दो। ये साला बेकार
हमें जो है ये जो तीन झावड़ा पकड़त ना, ये देख, ये साला भाई भाई कर रहा है सब, तो माइक वो है पकड़ेगा ही। अब इसको हम क्या करेंगे? इतनी आवाज सही है। हेलो हेलो है ना, एक सिंगल माइक चला, ठीक है। तो ठीक कराओ तब तो ठीक ना करेंगे।
बहुत स्लो चल रहा है।
नेट स्लो है, तो इसलिए आपकी आवाज रुक के आ रही है। जहाँ भी
यूट्यूब खोलने में क्या दिक्कत हो रही
नहीं
है?
तो क्या उसको बोल दो ना कि क्या नाम है, बोलो यूट्यूब भी चलने में इतना टाइम लगा रहा
फिर देखो नहीं आ रही है।
है। आवाज उठाई है?
नहीं उठा रही ना, यही तो बता रहे हैं कि अपने लोकल नहीं आ रही है। माइक पे चेंज करेगी नहीं, तो माइक पे ऑटो उठाइए बाई डिफॉल्ट। इसके लिए ठीक है,
ठीक है चलो हम बताते हैं।
हम बता रहे हैं कंट्रोल, तभी तो आवाज नहीं आ रही। हमारी आवाज आ रही है। दो लोग चार लोग हो तो कल वीडियो कॉल लगा के लाए हैं। कल वीडियो कॉल लगा कल वीडियो कॉल कल तो वहाँ लगेगा।
उसमें होता है ना, सुनती में।
कल उसमें
सुनती में यहाँ काम होता है, लेसन तो वहाँ होता है। फॉर्मेशन कन्वोकेशन सब उसी में होता है।
वहाँ वीडियो कॉल लग रहा होगा। हाँ लग जाएगा। हाँ आज ही लग जाएगा। अब अपना
हमारा पता
जूम भी तो है
हाँ, जो है।
कैमरा।
हमको नहीं पता ये सब, हम आज की ड्यूटी के जिम्मेदार है।
कल कौन है?
कल जिसकी होगी आप ही है।
और कौन है?
संदीप है, शिशिर है।
वो हीट कर रहा है ना, हीट पे कर रहा
चल रहा नहीं, एक घंटा तो हो ही गया, एक घंटा ज्यादा हो गया। प्रोजेक्टर बदलने वाले हैं।
है। हाँ भाई तो अब वो
ल्यूमिनेंस खत्म हो गया वो, या तो वहां की बजाय यहाँ कहीं लगाए।
हाँ बंद करो। सर्दियाँ नहीं आ रहा है,
दूसरी पे है।
हेलो 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 चेक 1 चेक हेलो 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 हेलो
करना कब है? आप क्यों?
देखो करेक्ट है, देखो देखो एक बार कनेक्ट कीजिए आप जरा, कनेक्ट करिए।
उधर से बोलिएगा।
अपडेट करना मांग रहे थे।
भगवत दर्शन देंगे।
हेलो।
हेलो हेलो।
हाँ सर, आ रही है आवाज?
आ रही है ना? ठीक है, मेरे
आ
पास
रही
भी
है।
आवाज आ रही है, आवाज आ रही है मेरे पास भी।
सुरेंद्र थोड़ी आवाज बढ़ा दो। हेलो, बोलते रहिए सर जरा।
हाँ, हेलो हेलो हेलो। माइक टेस्टिंग, हेलो हेलो।
हाँ ठीक है।
हेलो 1 2 3, हेलो 1 2 3।
ठीक 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 सर।
चलो
ओके।
ठीक है। ओके।
हरे कृष्णा 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 राम श्यामा 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 श्रीमन्नारायण नारायण बोलो राम
यही
राम।
बात है। देखना एक ऑप्शन नहीं तो नहीं दे रहे, रिमूव करना पड़ेगा।
हरे कृष्णा 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 हरे कृष्णा 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 जय श्री राधे 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 हरे कृष्णा 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 हरे कृष्णा 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 जय श्री राधे 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 हरे कृष्ण गोविंद हरे मुरारी हे दास नारायण वासुदेवा। कृष्ण गोविंद हरे मुरारी
आईडी बताओ। एक में तो वाइफ नहीं करना। आ गए जी, हाँ। बताओ आईडी बताओ। आईडी बताओ।
894 5627 1302 50 16 84 50 16 84। सामने वाले से जा रहा है।
हेलो
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 नहीं तो फॉलो वही को आना चाहिए था।
हेलो
क्या पार्टिसिपेंट बनना है? अच्छा कनेक्ट होगा
हेलो
यार।
माइक टेस्ट
ठीक है ना? एक मिनट। हाँ, हाँ।
चला जाए।
अरे यार हम तो सुबह ही बुला लिया था।
टाइम खराब करता
है
है।
ना? इसके जगह सोने हमने बच्चे को भी स्कूल नहीं छोड़ा। बताओ तो मैं छोड़ के आता। अब राजेश को बोलना था।
अभी तो कोई आया भी नहीं।
हम्म।
यहाँ पे दर्शन ही नहीं करने आए हैं। हमको लगता है 11 बजे से पहले कोई
11
नहीं
बजे से पहले कुछ छोड़े ही नहीं।
है। रिकॉर्डिंग का चल रहा है। रिकॉर्ड तो
हम कर नहीं कर सकते ना। हम वो नहीं है, वो तो होस्ट होना है।
होस्ट वो है।
तो शायद बता दिया।
स्टार्ट नहीं हुआ पास
तो ऑफ कर दिया ना।
पॉज पॉज हो गया। अब ध्यान रखना इसको।
तुमने फोटो नहीं भेजी तुमानसी से सारी ली।
यहाँ पे है जवानी सपोर्ट मांगो ना, क्यों नहीं देंगे? चेक हेलो हेलो वन चेक हेलो हेलो चेक हेलो 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 शुरू था क्या हो गया?
हो गया 9:30
नवा बजा
जब शुरू हो जाएगा तब समझ लो 9:30
चार्ज हो रहा है ना, लैपटॉप चार्ज हो रहा ना?
हाँ चार्ज हो रहा
बस
है।
ठीक,
हाँ हो रहा
बस ठीक
है
है।
लेकिन बहुत ज्यादा रुकता नहीं है। अगर
अरे
बंद
ठीक है बस इनका
प्लग बंद हुआ तो समझो सब पट है बंद हो। चल रहा तो सबसे
हरे कृष्णा 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 बोलो श्यामा 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 श्रीमन्नारायण नारायण बोलो रामा रामा हरे कृष्णा 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 बोलो श्यामा श्यामा श्रीमन्नारायण नारायण बोलो रामा कौन कहते हैं भगवान खाते नहीं वीर शबरी के जैसे खिलाते नहीं कौन कहते हैं भगवान खाते नहीं वीर शबरी के जैसे खिलाते नहीं अच्युता किशोर कृष्ण दामोदर राम नारायणं जानकी वल्लभम् काम करते चलो कृष्ण राम करते चलो बोलो राम बोलो श्याम हरे हरे कृष्ण सीताराम राम 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 श्याम बोलो श्याम बोलो श्याम 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 राम बोलो राम कौन कहते हैं भगवान सोते नहीं माँ यशोदा के जैसे सुनाते नहीं सोते नहीं माँ यशोदा के जैसे सुनाते नहीं अच्युतम केशवम कृष्ण दामोदरम राम नारायणं जानकी वल्लभम् याद आएगी कभी भगवान दर्शन देंगे कभी ना कभी हरे कृष्णा 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 बोलो श्यामा 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 श्रीमन्नारायण नारायण बोलो राम राम हरे कृष्णा 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 बोलो श्यामा श्रीमन्नारायण नारायण बोलो राम राम कहते हैं नाचते नहीं तुम गोपियों के जैसे नचाते नहीं तुम गोपियों के जैसे नचाते नहीं अच्युतम केशवम कृष्ण दामोदरम राम नारायणं जानकी वल्लभम् काम बिगड़े बनाएंगे वो तो सभी पार करेंगे मेरा कभी ना कभी हरे कृष्णा 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 हरे कृष्णा 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 जय श्री राधे 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 जपते मन हरे कृष्णा 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 हरे कृष्णा 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 जय श्री राधे 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 हरे मना श्री कृष्ण गोविंद हरे मुरारी हे नाथ नारायण श्री कृष्ण गोविंद हरे हे नाथ नारायण श्याम राधे 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 श्रीमन राधे राधे जपो राधे 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 श्रीमन राधे 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 जपो राधे 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 जपो कृष्ण दामोदरं राम नारायणं जानकी वल्लभं अच्युतं केशवं कृष्ण दामोदरं राम नारायणं जानकी वल्लभं कौन आते नहीं तुम मीरा के जैसे बुलाते नहीं आते आते तुम मीरा के जैसे बुलाते नहीं हरे कृष्णा 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 बोलो श्यामा 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 श्रीमन्नारायण नारायण बोलो राम राम हरे कृष्णा 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 बोलो श्यामा 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 श्रीमन्नारायण नारायण बोलो राम राम कौन कहते हैं भगवान खाते नहीं वीर शबरी के जैसे खिलाते नहीं कौन कहते हैं भगवान खाते नहीं वीर शबरी के जैसे खिलाते नहीं अच्युतम केशवम कृष्ण दामोदरं राम नारायणं जानकी वल्लभं नाम जपते चलो काम करते चलो कृष्ण राधा करते चलो बोलो राम बोलो श्याम हरे हरे कृष्णा सीताराम राम 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 श्याम बोलो श्याम बोलो श्याम 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 राम बोलो राम बोलो राम राम श्याम बोलो श्याम 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 कौन कहते हैं भगवान सोते नहीं माँ यशोदा के जैसे सुलाते नहीं कौन सोते नहीं माँ यशोदा के जैसे सुलाते नहीं अच्युतम केशवम कृष्ण दामोदरं राम नारायणं जानकी वल्लभं याद आएगी कभी भगवान दर्शन हरे कृष्णा 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 बोलो श्यामा 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 श्रीमन्नारायण नारायण बोलो राम राम हरे कृष्णा 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 बोलो श्यामा श्यामा श्रीमन्नारायण नारायण बोलो राम राम कहते हैं भगवान नाचते नहीं तुम गोपियों के जैसे नचाते नहीं नाचते नहीं तुम गोपियों के जैसे नचाते नहीं अच्युतम केशवम कृष्ण दामोदरं राम नारायणं जानकी वल्लभं Krishna Hare Krishna 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 Jai Shri Radhe 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 Japre Mana Hare Krishna Hare Krishna 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 Jai Shri Radhe 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 Japre Mana.
Hey, hurry up तुम. तुम्हारी भी नहीं थी.
Krishna Ram Narayanam Janaki Vallabham Achyutam Keshavam Krishna Ram Narayanam Janaki Vallabham तुम मीरा के जैसे बुलाती नहीं तुम मीरा के जैसे हरे कृष्णा 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 बोलो क्षमा 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 श्रीमन्नारायण नारायण बोलो राम राम हरे कृष्णा 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 बोलो श्रीमन्नारायण बोलो राम राम. कौन कहते भगवान खाते नहीं वीर शबरी के जैसे खिलाते नहीं वीर शबरी के जैसे खिलाते नहीं अच्युतम केशवम कृष्ण राम नारायणं जानकी वल्लभम् नाम जपते चलो काम करते चलो कृष्ण नाम करते चलो बोलो राम बोलो श्याम हरे हरे कृष्ण सीता के जैसे सुनाते नहीं सुनते नहीं माँ यशोदा के जैसे सुनाते नहीं अच्युतम केशवम कृष्ण दामोदरम राम नारायणं जानकी वल्लभम् याद आएगी कभी भगवान दर्शन देंगे कभी ना कभी हरे कृष्णा 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 बोलो श्यामा 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 श्रीमन्नारायण नारायण बोलो राम राम हरे कृष्णा 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 बोलो श्रीमन्नारायण नारायण राम राम कहते हैं नाचते नहीं तुम दिलों के जैसे नचाते नहीं तुम गोपियों के जैसे नचाते अच्युतम केशवम कृष्ण दामोदरम राम नारायणं जानकी काम बिगड़े बनाएंगे वो तो सभी पार करेंगे मेरा कभी ना कभी हरे कृष्णा 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 हरे कृष्णा 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 जय श्री राधे 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 जप रे मन हरे कृष्णा 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 हरे कृष्णा 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 श्री कृष्ण गोविंद हरे मुरारी हे नाथ नारायण वासुदेवा श्री कृष्ण गोविंद हरे हे नाथ नारायण श्याम भगवान मेरे राधे 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 श्रीमन राधे जपो राधे 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 श्रीमन राधे राधे जपो राधे 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 जपो राधे 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 श्रीमन राधे राधे अच्युतम केशवम कृष्ण राम नारायण अच्युतम केशव कृष्ण दामोदरम राम नारायणं जानकी वल्लभम् अच्युतम केशव कृष्ण दामोदरम राम कृष्ण कृष्ण बोलो क्षमा 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 श्रीमन्नारायण नारायण बोलो राम राम हरे कृष्ण 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 बोलो क्षमा क्षमा श्रीमन्नारायण नारायण बोलो राम कौन कहते हैं खाने खाते नहीं वीर शबरी के जैसे खिलाते नहीं कौन कहते हैं खाने खाते नहीं वीर शबरी के जैसे खिलाते नहीं अच्युतम केशव कृष्ण राम नारायणं जानकी वल्लभम् नाम जपते चलो काम करते चलो कृष्ण राधा करते चलो बोलो बोलो श्याम हरे हरे कृष्ण सीताराम बोलो राम 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 श्याम बोलो श्याम बोलो श्याम अच्युतं केशवं कृष्ण दामोदरं राम नारायणं जानकी वल्लभम् याद आएगी कभी हरे कृष्णा 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 बोलो क्षमा 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 श्रीमन्नारायण नारायण बोलो राम राम हरे कृष्णा 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 बोलो क्षमा क्षमा श्रीमन्नारायण नाचते नहीं तुम गोपियों के जैसे नचाते नहीं कृष्णा जय श्री राधे 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 जपते मन हरे कृष्णा 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 हरे कृष्णा 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 जय श्री राधे 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 जपते मन श्री कृष्ण गोविंद हरे मुरारी हे नाथ नारायण श्री कृष्ण गोविंद हरे हे नाथ नारायण श्याम राधे 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 श्रीमन राधे 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 यमो राधे 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 श्रीमन जपो राधे 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 यमो राधे 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 श्रीमन राधे राधे अच्युतं केशवं कृष्णदामोदरं रामनारायणं जानकीवल्लभम् अच्युतं केशवं कृष्णदामोदरं रामनारायणं अच्युतं केशवं कृष्णदामोदरं रामनारायणं जानकीवल्लभम् अच्युतं केशवं कृष्णदामोदरं रामनारायणं जानकीवल्लभम् कौन कहते हैं आते नहीं तुम मीरा के जैसे बुलाते नहीं कौन कहते हैं आते नहीं तुम मीरा के जैसे बुलाते नहीं हरे कृष्णा 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 बोलो श्यामा 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 श्रीमन्नारायण नारायण बोलो राम राम हरे कृष्णा 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 बोलो श्यामा श्यामा श्रीमन्नारायण नारायण बोलो राम राम कौन कहते हैं भगवान खाते नहीं वीर शबरी के जैसे खिलाते नहीं कौन कहते हैं भगवान खाते नहीं वीर शबरी के जैसे खिलाते नहीं अच्युतम केशवम कृष्ण दामोदरम रामनारायणं जानकीवल्लभम् नाम जपते चलो काम करते चलो कृष्ण नाम करते चलो बोलो राम हरे कृष्ण कृष्ण दामोदरम राम नारायणं जानकी वल्लभम् याद आएगी कभी हरे कृष्णा 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 बोलो श्यामा 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 श्रीमन्नारायण नारायण बोलो राम राम हरे कृष्णा 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 बोलो श्यामा तुम गोपियों के जैसे नचाते नहीं तुम गोपियों के जैसे नचाते नहीं अच्युतम केशवम कृष्ण दामोदरम राम नारायणं जानकी वल्लभम् काम बिगड़े बनाएंगे वो तो सभी पार करेंगे लीला कभी ना कभी हरे कृष्णा 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 हरे कृष्णा 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 जय श्री राधे 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 जपने मनाए कृष्णा 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 हरे कृष्णा 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 जय श्री राधे 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 हरे मना श्री कृष्ण गोविंद हरे मुरारी हे नाथ वासुदेवा श्री कृष्ण गोविंद हरे मुरारी हे नाथ राधे 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 श्रीमन राधे 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 यपो राधे 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 श्रीमन राधे 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 जपो राधे 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 यपो राधे 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 श्रीमन राधे राधे अच्युतं केशवं कृष्ण दामोदरं राम नारायणं जानकी वल्लभम् अच्युतं केशवं कृष्ण दामोदरं राम नारायणं जानकी अच्युतं केशवं कृष्ण दामोदरं राम नारायणं जानकी वल्लभम् अच्युतं केशवं कृष्ण दामोदरं राम नारायणं जानकी वल्लभम् कौन कहते हैं आते नहीं तुम मीरा के जैसे बुलाते नहीं कौन कहते हैं कृष्ण कृष्ण बोलो श्यामा 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 श्रीमन्नारायण नारायण बोलो राम राम हरे कृष्ण 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 बोलो श्यामा 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 श्रीमन्नारायण बोलो राम राम कौन कहते हैं भगवान खाते नहीं वीर शबरी के जैसे निभाते नहीं कौन कहते हैं भगवान खाते नहीं वीर शबरी के जैसे निभाते नहीं अच्युतम केशवम कृष्ण दामोदर राम नारायणं जानकी वल्लभम् नाम जपते चलो काम करते चलो कृष्ण राधा करते चलो बोलो राम बोलो श्याम हरे हरे कृष्ण सीताराम राम 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 बोलो श्याम बोलो श्याम 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 राम बोलो राम बोलो श्याम 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 कौन कहते हैं भगवान सोते नहीं माँ यशोदा के जैसे सुनाते नहीं कौन सोते नहीं माँ यशोदा के जैसे सुनाते नहीं अच्युतम केशवम कृष्ण दामोदरम राम नारायणं जानकी वल्लभम् याद आएगी कभी हरे कृष्णा 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 बोलो श्यामा 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 श्रीमन्नारायण नारायण बोलो राम राम हरे कृष्णा 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 बोलो श्यामा श्रीमन्नारायण नारायण राम कहते हैं भगवान नाचते नहीं तुम गोपियों के जैसे नचाते नहीं नाचते नहीं तुम गोपियों के जैसे नचाते नहीं अच्युतम केशवम कृष्ण दामोदर
Thank you very much for the opportunity and invitation. President of World PGI's for annual research day. It's a real pleasure and honor to be here with colleagues and friends, and the place we have spent very important 4 years of the customer, years of our life, and to get to the point and to be able to do something meaningful in life. So thanks so much for this opportunity. I want to talk something about a project that we have going on in the lab for quite some time. It was more about modifying T cells, where you can modify their genes and target tumors, and we have been able to, at least in this case, take some of the research from the lab and
सुरेंद्र, सरस्वती बनना है ना? नहीं है? चला दोगे ना?
से हाँ सर हम ये अंदर है थिएटर में बस सर आ रहे हैं ये वो हाँ हाँ अच्छा अच्छा आ रहे हैं
राष्ट्रीय सेवा से सारे अपडेट्स पाने के लिए बेल आइकन दबाएं।
क्योंकि ग्रुप में भेज दी गई थी सर सर वाली जो नोटिस को मजेदार देखने के बाद भी सेट करके
अरविंद जी डाउनलोड नहीं हो रहा है ये हाँ एसएस लिखा था तो यूट्यूब बहुत धीमे चल रहा है स्लो है नेट
राष्ट्रीय सेवा से सारे अपडेट्स पाने के लिए बेल आइकन दबाएं।
क्या कह रहे हो यहाँ पे ये डब्ल्यू पहले या बाद में फिर स्लैश नहीं हो रहा सिक्योरिटी रीजन सिक्योरिटी रीजन किया है ये बात
टाइम्स पे कर देते हैं
इसमें होगा कहीं कहीं तो किया था
सर्च करके देखा अच्छा उसकी
नहीं वो तो हो चुका है
इसका टेम्पररी फाइल
जनगण मन आ गया देखो इससे सब करना जनगण मन आ गया इससे सब करना पता नहीं देखो पहले क्या पता चलो देखो
इसका टेम्पररी फाइल डिलीट करो सर पता नहीं
Rakshi wants to start your base farming thing, but I can do fine.
यहाँ करेंगे ना ये है ना
डॉट लिखा था
डॉट के आगे
नहीं लिखा था वो आके मना कर दे रहा है सिक्योरिटी रीजन ये देखो
प्राइवेट से ब्लॉक कर रखा है नहीं हमने डाउनलोड करने की परमिशन बंद कर रखा होगा पीजीआई वाला क्लाइंट है ना नहीं देखिए बंद कर रही है ना आईपी से ब्लॉक कर रखा है तो ये होगा नहीं तो एक काम कर सकते हैं
इसको मिनिमाइज करके रख दो
नेट नहीं चल रहा उसमें तो
कहाँ से डाउनलोड कर लोगे
मोबाइल पे काम करना उसमें बीच में ऐड आ जाएगा तो क्या करोगे
नहीं प्ले कर लो उतना बस वहीं से ऐड के बाद स्टॉप कर दो
नहीं वो बात नहीं है बीच में भी ऐड आ जाएगा
नहीं आएगा बीच में
Rakshi
ये
wants to
खत्म हो
start
जाए
your
बस
base
इसको
farming thing, but I can do
जैसे
fine.
वो आएगा बस अब स्टॉप कर दो बस
बफरिंग हो रही है ना
नहीं डाउनलोड होगा तो क्या करोगे
बता दो जनगणना तो मिल गया आपको
टेम्पररी फाइल अच्छा डिलीट करो पहले आराम से इसको स्लो चल और जनरल में नहीं चलाना
भी नहीं चल रहा है ना जनगणना भी नहीं चल रहा है मैं देख रहा था
चलिए हम तो बोलते लेके आओ।
बोलो जाकर
सारे प्रोग्राम बंद कर दो, जियो भी बंद हो जाएगा।
नहीं
हैं? और यहाँ से खाली कर दो। अरे
ये विजयनगर में नहीं
हम नहीं।
बुलाओ।
सुनिए,
जनगण मन अधिनायक
इसको नहीं छेड़ना है।
तो देख लीजिए ना, ये आपकी इच्छा
कहाँ
में
रखा है?
यही तो खोज रहा है ना।
ये है तो सरस्वती का नाम।
ये देखो ये खोज रहा है ना, ये मिल रहा है ना
ये है तो यही तो है। हाँ, ठीक है ठीक है।
आवाज दो।
आवाज बस ठीक है। ठीक है कहाँ है? उसको रिसर्च में डाल दो।
हो गया पता।
रिसर्च इसमें डाल दो। इसका रीनेम कर दो जरा, ये जो इसका रीनेम है डॉक्टर शिखर मल्होत्रा। डॉक्टर शिखर
किसने रखा है? अरे क्या जबरदस्ती है।
6 जनरल मन भी रख लो इसको भी। करके रख लो।
देखा तो क्या करोगे?
रख लो।
मिल गया ना तो
इसमें डाल दो। ये डेस्कटॉप खाली कर दो, वो पीपीटी हटा दो सब। इससे वीडियो को छोड़ के बाकी सब यहाँ क्यों इसी में ही आएगा तो यहाँ बनाओगे।
नहीं खोल नहीं मिल रहा है।
पहले ये खाली करो दूसरे को दो।
ये कंडक्टर क्या लिखा है?
अरे अच्छा नहीं, ये आप सब जितने हैं खाली रजिस्टर देखो छोड़ के बाकी जितना है सब डी में ले जाके कॉपी करते हैं।
इसमें रिसर्च पोस्टर फाइनल ये वाला नहीं,
सिर्फ इसी में था।
ये तो पोस्टर्स का है ना सिर्फ। एक रिसर्च डे का फोल्डर बनाया था ना, इसी में खोलिए फिर। इसी में सारा
खोलिए था। हम्म,
ये जो
हाँ सर, दो हैं, दो हैं।
एक फैकल्टी को
हाँ,
स्टूडेंट।
एक बार आप ये ऊपर फैकल्टी का दोनों ओपन कर दीजिए। हाँ, एक ही ओपन करिए।
ये पोस्टर है।
हाँ हाँ, ओपन करिए।
हाँ ये वाली एक लिस्ट और इसके नीचे वाली है, ये मेल हो पाएगी
मेनू चाहिए।
नहीं तो सर
या व्हाट्सएप पे शेयर हो पाएगी।
या हाँ, आप वो इंटरनेट है। आप
इधर
एक
बहुत
सेकंड
स्लो
आप
है,
मुझे दे दीजिए।
मुझे
स्लो है।
बेवक्त दे दो ना, इसे उसके बाद मेरा नंबर नहीं होगा ना तुम्हारे पास।
यस सर।
नहीं, लेकिन फिर मैं अपने व्हाट्सएप से अपने आप को ही भेज लेता हूँ ना।
आपको स्कैन कर
हाँ हाँ
इंटरनेट स्लो है, चाहिए इसका है ना तो सर हमको जूम चलाने के लिए दिया है। क्या जूम चला सकते
स्कैनर
है सर थोड़ा सा।
नेशनल एंथम शुरू में भी होगा, लास्ट में भी होगा।
लास्ट में भी
नहीं, शुरू में भी वो कुछ बता रहे हैं। कंपटीशन में जैसे शुरू में भी होता है।
कर लिए इसका। यूजुअली शुरू में तो वो क्या बोलते हैं
सरस्वती बननी है।
सरस्वती वंदना वगैरह।
देखो कोई बाहर होते नहीं।
मैम हमको यहाँ का कौन है पता नहीं,
अंदर वाला नहीं होते।
यहाँ कोई नहीं।
टाइम लगेगा।
उसको भी मिनिमाइज कर देते हैं। दोनों शीट टैब कॉपी कर देंगे पीडीएफ।
हाँ मैं संतोष भाई, बस मैं वही फाइल ले रहा हूँ। ऑडिटोरियम में अंदर हूँ। वो जो फाइनल एक्सेल फाइल जो है वही ले रहा हूँ मैं। अभी वही देख रहा हूँ यार।
अटैच करना पड़ेगा। कॉपी करके रख सकते पेस्ट कर लीजिए।
यस अब एक कई दो
लॉग आउट
हाँ,
से लॉग आउट।
तो खैर डिवाइस से भी हो
हाँ डिवाइस से कीजिए इससे।
थैंक यू
यस वेरी गुड। थैंक यू सो मच।
हेलो चेक मान चेक। हाँ क्या
पीपीटी खोलो
पीपीटी एक ही तो आई है।
खोलो तो
कोई यहाँ की भी आई भी नहीं। जो लेक्चर लाइव वाला था उसका है, उनका कोई नहीं आया। आई तो थी, पता नहीं कहाँ है। उसी की फोटो अरे यही नहीं हट रहा।
बस यहीं लगा बंद है।
ये अरविंद जी दिखाया नहीं। मैंने कर दिया नहीं।
कहाँ डाल दूँ? कहाँ डाले? सेल्फ में जाओ ना,
सब में
अपना नंबर डाल रहा है, वहाँ पे नहीं है। क्या नाम डालो?
नाम डालो।
अपना नाम डालो ना, बबलू है ना, ये लिखो। आया? ये वाला नीचे वाला है ना,
जी
ये तो आपको भेजा नहीं होगा आपको। एक बार तो गंगा बलम डाल दो ना, जैसे तो
जब मुझे भेज देते किसी में भेज देते और खाना चाहना आपका डॉक्यूमेंट चला जाता है।
बंद कर दिया।
अपना क्या था डॉक्यूमेंट था ना। बी बी ए यू दिखाते हैं। क्लोज कर देते हैं। जरूरत क्या है। चालू करके हेलो 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 हेलो
हेलो हेलो साउंड तो शुरू
हाँ जी एक बार ओपन कर दीजिए। नहीं ठीक है, हो गया।
ये आप रहेंगे ना, ये फोल्डर बना दीजिए।
ठीक है।
इसमें ये सब करिए। उसके बाद शायद पहला लेक्चर ये है।
पहला वीडियो का
पहला इनका
क्लास
अच्छा वीडियो लेक्चर लग रहा है। ठीक है। अभी एक कोई और वीडियो है।
हाँ एक और बच्चे
देखिए अलग हो जाएगा।
हाँ
Otherwise, if you have a pen drive, I can copy again this pen drive my computer. So it's not नहीं कर रही। वायरस this one. Oops, I am the guy. I'm copying. Checking the it's you have another pen drive? मैं अभी एक कॉपी देता हूँ।
मैं एड्रेस करूंगी, आप आके क्या करोगे ना?
बस बुला देना, बाकी ये मैं इंट्रोडक्शन में दे दूंगा। उधर से डिलीट हो सर हटा।
सर जब बोलिएगा तो उसको ऑन कर लीजिएगा, ये बटन लगा हुआ है, उसको पुश करिएगा ऑन हो जाता है। यही दबा के अभी ऑफ है, जब पुश करेंगे तो इसमें लाइट जल जाएगी। एसी कदर चल रही है ना तो आवाज लग रही है, तो इसमें हमिंग हो रही है।
Research is the heartbeat of progress, echoing the curiosity that drives us to explore the unknown. Good afternoon to all the dignitaries, delegates, and my dear colleagues. Welcome to 4th Research Day of Sanjay Gandhi Postgraduate Institute of Medical Sciences. It gives me immense pleasure to extend you a very warm welcome on behalf of the organizing committee, who has clubbed all of us for the SGPGI Research Day 2023. I, Arunim Shah,
I, Ananya Bajpayee, and we are delighted to be your anchor for today's exciting event. Today, we have gathered here to celebrate and showcase the incredible research work conducted by our brilliant minds here at SGPGI. As we dive into the fascinating world of research, we are about to witness groundbreaking ideas, innovative solutions, and a wealth of knowledge that will undoubtedly leave us inspired and amazed. I welcome Honorable Director of our Institute, Professor R.K. Dhiman. I would welcome Dean of our Institute, Professor Shalin Kumar.
Now, I kindly request Dr. C.P. Chaturvedi to escort the dignitaries to the dais. And I thank all the eminent personalities who have spared time to enlighten us with their knowledge to make this day a wonderful learning experience. I would like to request Dr. C.P. Chaturvedi to present a token of gratitude to our Honorable Director, Dr. Professor R.K. Dhiman. As per our institute's tradition, all academic activities begin by recitation of Saraswati Vandana and lamp lighting. Now, I humbly request all the dignitaries to come forward and light the lamp before Ma Saraswati. I would also request the eminent dignitaries, Dr. Gautam Palid, Dr. V.P. Sharma, and Dr. Chandeshwar Nath to join
Now, following this pleasant illumination, I would like to invite Dean Professor Shalin Kumar to extend his welcome to all the dignitaries.
Director Dhiman Sahab, our distinguished judges today, and also luminaries in their fields, particularly Dr. Gautam Palit, who is the ex-head of Department of Pharmacology and Senior Deputy Director of CSIR CDRI Lucknow, Dr. V.P. Sharma Sahab, Senior Scientist, and Dr. Chandeshwar Nath Sahab, who's also very prominent in our ethics committee and have contributed. There are so many others here who've been invited, distinguished judges, faculty members, colleagues, students, ladies and gentlemen. Why research in a teaching hospital? To any doctor, any healthcare professional who's worth his or her salt will always ask the question, is this the best I can do? Can I do better? Why did this patient not respond? Why did this therapy work? Could it be cheaper? Could it be safer? Could it be more effective? In order to answer this question, which should be every single healthcare professional's job, you engage in a scientific, predefined, well thought out process where you assemble data, do some analysis, see what the probabilities are, and then do prospective studies. And then you design research questions appropriately, whether these be in the context of basic research, whether they be in the so-called applied research, or whether they be in the context of direct patient care. It does not matter. What matters in the end is the quality of work that you do. Is it a useful addition, or is it just one more addition? We've had reasonable attendance in the reasonable number of posters presented today, a fair amount of enthusiasm. Things could have been slightly better, and I would request colleagues and attendees to provide a feedback of how we can make this process better, more interactive, and how can we just improve on what we are doing. And thank you all for your participation. And with that, I think we will proceed with the rest of the day, with the rest of the function. And we request now our director, sir, to please, enlighten us with his views on, how and what research should be for us in much more detail and other things he deems appropriate. I thank you all for your attention.
Thank you, sir, for such a warm welcome. Now I would like to invite Director, Sir, Professor Deeman, to address the event.
Thank you and good afternoon. Professor Shalin Kumar, Ovadi, Professor Sanjay, our Chief Medical Superintendent, Dr. C.P. Chaturvedi, Faculty in Charge Research, distinguished judges, my dear colleagues, ladies and gentlemen. आज हम जो हैं 4th रिसर्च डे मना रहे हैं। अगर आप देखें इंस्टीट्यूट की जो हिस्ट्री है, इट डेट्स बैक टू 1984, जब इसका शिलान्यास किया गया था। उस समय के प्रेसिडेंट साहब थे नीलम संजीवा रेड्डी। 1987 में इस इंस्टीट्यूट का क्लिनिकल, जिसको कहना चाहिए पेशेंट केयर, टीचिंग एंड ट्रेनिंग स्टार्ट हुआ। तब से लेके 87 से लेके अगर हम अभी तक की बात करें तो 35-36 साल हो गए हैं, लेकिन हम रिसर्च डे आज चौथा ही मना रहे हैं। अगर आप पीजीआई का जो लोगो है उसको देखें तो उसमें 3 पिलर्स हैं। फर्स्ट पिलर इज फॉर द पेशेंट केयर, and the second pillar is for the teaching and the training, and the third is very important pillar, that is the research board and institute. अगर आप देखते हैं कि इंस्टीट्यूट का जो स्थान है, NIRF में 7th है, नॉर्थ इंडिया में 3rd है आफ्टर द एम्स एंड द पीजीआई चंडीगढ़. It is not only because of the patient care and the teaching and training, but a major contribution by the research also. That is very important. अगर हम टीचिंग एंड ट्रेनिंग की बात करें तो हमारे यहाँ कॉन्वोकेशन होता है। पेशेंट केयर की बात करें कि हमने कौन कौन सी फैसिलिटीज बढ़ाई हैं, किस ग्रुप के पेशेंट आ रहे हैं, रोबोटिक स्टार्ट किया है या ओटी स्टार्ट किए हैं या गामा नाइफ स्टार्ट किया है, दैट कम्स अंडर द फाउंडेशन डे। लेकिन हम रिसर्च की बात करें तो अलग से कोई दिन नहीं है। मैं तो कहूँगा कि जो रिसर्च डे है ये रिसर्च का एक फेस्टिवल है, महोत्सव है। और आप जजेस साहब भी हमारे डिस्टिंग्विश्ड रिस्पेक्टेड जजेस आए गए हैं, तो हम लोगों ने देखा भी होगा एक फेस्टिवल ही आज दिखाई दिया हम लोगों को। मैं हर लोगों से मिला हूँ तो सर तो हमको बोल रहे थे कितना हाई स्टैंडर्ड है यहाँ का। इट इज़ वेरी डिफिकल्ट टू फाइंड जो रिसर्च यहाँ पर हुई है। मैंने भी पूरा देखा वहाँ पे, तो गुड इम्पैक्ट फैक्टर जर्नल्स में इनके पब्लिकेशन हो चुके हैं और कई रिसर्च पेपर्स तो खाली पब्लिकेशन के लिए नहीं है, वो एक हमारी सोसाइटी के लिए भी है, कई ऐसे हमने देखे। और सबसे ज्यादा खुशी मेरे को तब हुई जब मैंने नर्सिंग कॉलेज के पोस्टर्स देखे, they were published in journal with impact factor more than 6, and this was from our principal Dr. Radha K, बैठी हैं हमारे साथ में। तो जो मोटिवेशन जो है हमारे फैकल्टीज देते हैं, वो मोटिवेशन जो है वो वहीं पे खत्म नहीं होता, इट स्प्रेड्स आउट। जैसे लाइट होती है ना, द मोमेंट यू जस्ट और स्विच ऑन करते हो बल्ब का, तो आपका का सारा जो है थिएटर भी देख रहे हैं, पूरा का पूरा एनलाइटन हो जाता है। तो फैकल्टी भी अगर ऐसी चीज करते हैं तो इट एनलाइटन्स ऑफ द नेक्स्ट जनरेशन, our younger colleagues, our students. That's what we want at the moment. मैं देख रहा हूँ यहाँ पे बहुत सारे स्टूडेंट्स हैं, मैं पोस्टर्स के देख रहा था कि स्टूडेंट ने more than 170 पोस्टर्स लगाए हैं। ये बहुत बड़ी बात है। हर 3 में से एक स्टूडेंट ने पोस्टर लगाया। पिछले 4 रिसर्च डे हमने स्टार्ट करे हैं। पहला रिसर्च डे जब मैंने ज्वाइन किया था 2020 में तब हुआ था, उससे लेके मैं आज प्रोग्रेस देख रहा हूँ, हम 250 तक पहुंच गए। मैं उम्मीद करता हूँ नेक्स्ट टाइम इट विल बी मोर देन 300 और 350 और 400। इतने पोस्टर प्रेजेंटेशन एक नेशनल लेवल की कॉन्फ्रेंस में भी नहीं होते हैं। अगर आप देखें पकड़ पकड़ के मंगाए जाते हैं, चाहे वो हेपेटोलॉजी की हो, किसी भी कॉन्फ्रेंसेज हो जो नई ब्रांचेस हैं। लेकिन हमारे यहाँ अगर पीजीआई में ऐसा हो रहा है, so it is really a festival to celebrate, and we are celebrating it. And I take this moment to thank all my faculty, younger colleagues, students, residents, and everyone, including the nursing college and the College of the Medical Technology also. तो आज मैं तो अक्सर तो मैं एकेडमिक्स देखता ही रहता हूँ, पोस्टर देखता ही रहता हूँ, ग्रेडिंग करता ही रहता हूँ, लेकिन आज जो मैंने नया चीज मैंने देखी, जो जो मैंने अनुभव किया, जो मैंने फील किया, फील इज द राइट वर्ड, कि जो लोग प्रेजेंट कर रहे थे वो अपने आप को बहुत गौरवान्वित महसूस कर रहे थे, बहुत अच्छा लग रहा था उनको कि हम इस उसमें हैं. दैट्स व्हाट वी वांट एक्चुअली. It is not, I did not feel that it was a compulsion for our faculty or the residents to or the students to present the poster. मेरे को आज लगा उन्होंने अपने मन से ही जैसे आके ये काम किया है. सो दिस इज अ ग्रेट थिंग. और मैं आप लोगों को इस बात की पुनः बार बार बधाई देता हूँ. और बताना चाहूंगा रिसर्च के बारे में. जब से मैं आया हूँ मैंने कोशिश करी है रिसर्च आगे बढ़े. और मैं तो यहाँ तक भी कहता हूँ कि पेशेंट केयर, टीचिंग, ट्रेनिंग और रिसर्च जो है खाली बड़े बड़े इंस्टीट्यूट्स में नहीं, मेडिकल कॉलेज एवं डिस्ट्रिक्ट हॉस्पिटल में भी होनी चाहिए. सबसे सिंपल रिसर्च कैन बी परफॉर्म इवन एट द पीएससी. जो आप पेशेंट्स देख रहे हो उनका रिकॉर्ड रखो, उसको एनालाइज कर दो. तो द डाटा दैट यू रिसीव फ्रॉम द पीएससी, द पेरिफेरल फ्रॉम द विलेजेस एंड द अदर थिंग, it is really a very useful thing for our planning for the future also. तो रिसर्च को हमने प्रमोट किया. हमारे नंबर ऑफ द पीएचडी स्टूडेंट्स बढ़े. डिपार्टमेंट में जो रिसर्च हो रही थी वो बढ़े. उसका जो आंकलन हम लोग करते हैं कि रिसर्च बढ़ रही है नहीं बढ़ रही है. नंबर ऑफ द इंट्राम्यूरल प्रोजेक्ट्स दे आर इंक्रीजिंग बाय ईयर. अभी मेरे को पता चला कि लास्ट ईयर हमने इंटरम्यूरल 51 दिए हैं और एक्स्ट्रा म्यूरल की जो ग्रांट है वो तो 15-20 करोड़ में चल रही है यहाँ पे. और पहली बार से जितनी पहले थी 3-4 साल पहले उससे ज्यादा है. तो एक बहुत इंपॉर्टेंट बात है अपने आप में. जस्ट टू प्रमोट द रिसर्च एट द डिपार्टमेंटल लेवल we have increased the grant, intramural grant from the 5 lakhs to the 10 lakhs also. And ये बहुत बड़ी बात है कि 5 लाख में कई प्रोजेक्ट नहीं हो पाते थे तो 10 लाख में वो आराम से हो सकते हैं. मैं आपको बताऊं जब मेरी पहली आईसीएमआर ग्रांट मिली थी way back in 1996, it was only for the 6 lakhs. आज मैं देखता हूँ यहाँ पे करोड़ों करोड़ों में ग्रांट मिलती है. लेकिन वो 6 लाख और 10 लाख अगर आप कोई भी पायलट प्रोजेक्ट करना चाहते हैं, शुरुआत में देखना चाहते हैं, वो बहुत इंपॉर्टेंट है. कई डिपार्टमेंट ऐसे हैं जहाँ पे उन्होंने इनोवेशंस की हैं. रिसर्च इज नॉट ओनली इम्पोर्टेंट बट ऑल्सो द इनोवेशंस ऑल्सो. तो इसी उपलक्ष्य में मैं आपको बताना चाहूंगा इस बार 3 प्रेसिडेंट के अवार्ड हमने स्टार्ट करेंगे. एसजीपीजीआई प्रेसिडेंट्स अवार्ड. पहला तो वो है डिपार्टमेंट जिसने मैक्सिमम इंटरम्यूरल ग्रांट ली है. दूसरा वो है फैकल्टी जिसने मैक्सिमम एक्सटर्नल ग्रांट्स ली है या कि मैक्सिमम मनी वो लेके आए ग्रांट के उसमें. तीसरा है पेटेंट का. और मेरे को बड़ी खुशी हो रही है बताते हुए कि हमारे 2 फैकल्टी मेंबर्स दे आर गेटिंग द अवार्ड फॉर द पेटेंट्स आल्सो डे आफ्टर टुमारो ऑन द ओकेजन ऑफ द कन्वोकेशन डे. तो रिसर्च जो है मैं इस समय बोल रहा हूँ कि रिसर्च मस्ट कल्मिनेट इनटू अ पॉजिटिव थिंग. आप करिए, आप इनोवेशन करते हैं. लोगों ने वीडियो लेरिंगोस्कोप किया है, कई चीजें करी हैं और जो कि बहुत इंपॉर्टेंट चीजें हैं. और उसका पेटेंट आपने लिया है, बहुत इंपॉर्टेंट चीजें हैं और ये इंस्टीट्यूट के लिए बहुत इंपॉर्टेंट है. हमारे यहाँ खुद एक इंक्यूबेशन सेंटर है, SGPGI है, जिसमें कि हम कितने भी जो है स्टार्टअप स्टार्ट करते हैं. मैं आपको बताना चाहूंगा कि 18 तो फाइनल स्टेजेस में, 25 और आ गए हैं. लक्ष्य हमारा 50 का है. So there is lots and lots of the facility now. Not only doing the good research also, but you can have a patent also. And for this we have also opened at SGPGI the patent cell also. नहीं तो उनको इधर उधर जाने में प्रॉब्लम होती थी. So ladies and gentlemen, हम लोग ने पूरी कोशिश कर रहे हैं कि रिसर्च जो है यहाँ पे वर्ल्ड क्लास हो, कटिंग एज हो और जो हमारी प्रॉब्लम्स हैं प्रॉब्लम ओरिएंटेड हो. और आप लोगों ने बढ़ चढ़ के इसमें हिस्सा लिया है. ईयर वाइज देखेंगे प्रोग्रेस हो रही है. मैं आशा करता हूँ आगे ऐसा होता रहेगा. और आपके जितने भी आप लोगों ने इसके लिए प्रयत्न किए हैं, आई एम थैंकफुल टू ईच वन ऑफ यू. थैंक यू वेरी मच.
Thank you so much, sir, for the words of inspiration. Thank you. Moving forward with our scientific sessions, for the first keynote lecture, I would extend a warm welcome to Dr. Arun Kumar Trivedi.
Dr. Arun Kumar Trivedi is Professor ACSIR and Senior Principal Scientist at Central Drug Research Institute, Lucknow. He did his PhD from Ludwig Maximilian University, Munich, Germany, and postdoc research at the State Center for Cell and Gene Therapy, Saale, Germany. He is a recipient of prestigious individual German fellowship, Deutsche Hose, Carreras, and various awards, including Lady Tata Memorial Trust, Young Research Award, CSIR Young Scientist Award in Biological Sciences, INSA Medal for Young Scientists, NASI Young Scientist Platinum Jubilee Award, UPCST Young Research Scientist Award. He has more than 60 publications in reputed international journals to his credit. The primary focus of his lab is to understand mechanisms underlying breast cancer pathogenesis and impaired differentiation in acute myeloid leukemia. Today, he'll be delivering a lecture on mechanism of functional inactivation of CBP alpha leading to differentiation arrest in acute myeloid leukemia. Over to you, sir.
Thank you very much for the kind introduction. At the outset, I would like to thank organizing committee, particularly Dr. C.P. Chaturvedi, for inviting me to this gathering to deliver this lecture. Today, I shall be talking about mechanisms of functional inactivation of CBP alpha leading to myeloid arrest in AML. So, before I start to my key findings, let me briefly talk about what is differentiation arrest and why it is important. As you know, during hematopoiesis from hematopoietic stem cell, common myeloid progenitor and also side-by-side common lymphoid progenitors are formed. But because my findings are more on myeloids, I'm talking more about the myeloid pathway. So these common myeloid progenitor pathways then give rise to the megakaryocyte erythroid progenitors and granulocyte megakaryocyte progenitors, and then they subsequently differentiate to the functional cells. I don't think it is visible there quite nicely, but I think we will have to bear with it. What happens during leukemia is that because of the leukemogenic events, such as mutations or any chromosomal translocations, those blasts keep on accumulating. Here, these blast cells keep on accumulating, and once they are accumulated, the normal, completely differentiated cells are not formed, and because of this, there is differentiation arrest. And this differentiation arrest is often can be overcome by certain mechanisms, particularly ATRA differentiation therapy is there, particularly in APL cases where ATRA is given, and recently now for treatment point of view, IDH inhibitors are also there that can induce differentiation in these blasts so that these cells can be differentiated to the normal cells. Well, the standard form of therapy is the chemotherapy, but my point of showing this slide is that the key pathway of myeloid differentiation, particularly the formation of these, functional cells, is regulated by two key transcription factors, and they are CBP alpha and PU1. If there is higher amount of CBP alpha, lower amount of PU1, it drives towards the granulocytes, and if there is higher amount of PU1, lower amount of CBP alpha, it drives towards the macrophages. Now, in most of these cases, it has been shown that the function of CBP alpha is impaired. So, in all those cases where there is myeloid arrest, in majority of the cases, CBP alpha expression or function has been shown to be impaired. Now, what is CBP alpha? Well, CBP alpha is a transcription factor that contains a basic rich listen zipper, which is located in this C-terminal region, and it has two transactivation domains. So this BZ domain, it binds to the DNA, all its target genes, and through its transactivation domain, it interacts with the cofactors and co-repressors, and thereby it regulates number of target genes. Well, it is very important factor because it can regulate both differentiation as well as growth arrest. Through its transactivation domain, it binds with the cell cycle regulatory proteins, causes the growth arrest, and once the cells are arrested, cells can be, can exit from the cell cycle and they can differentiate to the functional cells. And there, while differentiation is taking place, it can regulate those factors that is required for the differentiation of the granulocytes. For example, GCSF receptor and CBP epsilon, these are the two key transcription factors that are regulated by CBP alpha and are required for the differentiation of these progenitors to the granulocytes. Well, CBP alpha was initially identified as a liver protein, but later on it was discovered that it is one of the key factors that is required for the formation of the neutrophils, particularly the granulocytes. Now, this was the landmark paper that was published in 1997 in PNS by Danter and group, where they showed that in a conditional knockout mice, when they evaluated all the blood lineage cell, particularly myeloid lineages, they found that even in the heterozygous, there were both neutrophils as well as macrophages, but in the conditional knockout mice, there were only monocytes and no neutrophils. So this was the first landmark study where they showed that CBP alpha has a key role in the formation of neutrophils. And down the line, it was found that in majority of the AML cases, CBP alpha is mutated. In almost 10 to 15% of the cases, there is mutations. And in many of the other cases, it has been shown that there are certain antagonistic proteins or post-translational modifications that make it inactive. Now, these are the other mechanisms how it has been shown that CBP alpha, I mean, like I said, it is very crucial for the neutrophil differentiation, and hence it is at the center of attack in most of the AML cases. Well, people have shown that in many of the cases there is CBP alpha mutations. There are also inhibitions at the mRNA and promoter level. There are promoter hypermethylations of CBP alpha, and particularly in case of CML, if you look at the BCR-ABL positive CML cases, there is CBP alpha mRNA, but there is no CBP alpha detectable protein. And people have shown one Nature Genetics paper where they showed that in BCR-ABL positive cells, there is no CBP alpha protein because the mRNA of CBP alpha is inhibited by the RNA binding proteins. So CBP alpha mRNA inhibition has been shown. Subsequently, if the protein is formed, people have shown that the post-translational modifications of CBP alpha occur that render it inactive. For example, phosphorylation at serine 21 in FLT3 ITD positive mutations makes it transcriptionally inactive. In addition, there are many antagonistic proteins that have shown to interact with it and inhibit its transcription ability. We have recently published many papers where we have shown that there are certain antagonistic proteins that can interact with CBP alpha and inhibit its function. Recently, we published some papers where we work particularly on the protein levels of CBP alpha, and there we try to identify what are the E3 ligases that might be involved in regulating CBP alpha protein stability. So that's what I will talk today about. But before I go into the protein stability pathway, let me briefly talk about the ubiquitin cascade. Of course, you all are aware, but I just would like to mention that ubiquitin cascade is basically a sequential action of various enzymes, E1, E2, E3, and very importantly, de-ubiquitinases drugs. So there is interplay between E3s. These are the E3s, and then these are the drugs. So what happens when a substrate, for example, CBP alpha in this case, is polyubiquitinated by these E3 ligases, and once that substrate is polyubiquitinated, that is subjected for the degradation by the proteasome pathway. However, simultaneously, there are certain dubs that keep on removing these ubiquitins so that the homeostatic levels of the protein is maintained in the cell. So there is utmost need of dynamic equilibrium between the action of E3s and dubs. So we hypothesize that if there are certain E3s that are activated during leukemia, and if they are polyubiquitinated in CBP alpha in a way that its protein levels are reduced, then that could be one of the possible target through which we can reactivate or restabilize CBP alpha. So today I'm going to talk about one E3 ligase and one kinase through which we have shown that they negatively modulate CBP alpha protein levels. Now, we in the first part, we are showing that CDK2, which is very common cell cycle kinase. I mean, one paper showed that during differentiation of blood cells through ATRA, because ATRA is a clinically used differentiation therapy in AML, and in that study that they published in 2018, they showed that during differentiation there is degradation of CDK2. So degradation of CDK2 is required for the induction of differentiation is what they're finding. So based on this, we speculated that if there is CDK2, it must be inhibiting CBP alpha, or it could be inhibiting CBP alpha. To address that, we first evaluated, does inhibiting CDK2 through pharmacological inhibitors brings any changes in CBP alpha protein levels. As you can see here, when we treated with the roscovitin, which is a CDK2 inhibitor, in THP1 and U937, these are both in vitro cell lines, there was restoration of CBP alpha in the treated condition, which indicated that inhibiting CDK2 might impact on CBP alpha protein levels. We further, in U937 itself, we depleted CDK2, and we found that when we depleted CDK2, there was copious restoration of CBP alpha in this condition. So these two, kind of ensured us that inhibiting CDK2 might restore CBP alpha. To address this further, we overexpress CDK2 to see if we are getting the reverse finding. And we found that when we overexpress CDK2 here, CBP alpha expression, the endogenous expression reduced. Similarly, because CDK2 was downregulating CBP alpha protein, and like I said, we were interested in looking at the protein level. So we evaluated if this downregulation is via proteasome pathway or not. So here, when we treated with the increasing amount of CDK2, you can see CBP alpha was going down. But when we inhibited with the proteasome inhibitor, in this case, if you compare between this and this, there was restoration, indicating that CDK2 might be downregulating CBP alpha by impacting its protein turnover. Now, to further confirm this, we perform this cyclohexamide treatment assay, where you inhibit the neoprotein synthesis, and whatever protein is available will degrade at its normal rate. So when we followed it up to 240 hours, we found that rate of degradation of CBP alpha was slower compared to when we overexpress CDK2. So the CDK2 expression exaggerated the degradation of CBP alpha. And this was further confirmed in also in the endogenous condition, that when we gave roscovitin, which is a CDK2 inhibitor, the CBP alpha protein was stabilized and its degradation was also restored. So indicating that CDK2 impacts CBP alpha protein levels. Now, like I said, that with proteasome inhibition, there was restoration of CBP alpha. So how it is doing this? Because CDK2 is a kinase, it is not an E3 ligase. But we wanted to confirm if it is really happening via ubiquitination. So we used here a mutant where all the lysins of the ubiquitins are mutated in this condition. So when we overexpress CDK2 along with CBP alpha, there was slight down regulation. But when we increase the amount of lysine mutant that was having all the lysins mutated in the ubiquitin, the CBP alpha protein rather stabilized, indicating that CBP alpha protein levels are modulated by ubiquitination. So this we further confirm that indeed, in the presence of CDK2, if we compare these two lengths, in the presence of CDK2, CBP alpha was heavily polyubiquitinated, indicating that CDK2 promotes CBP alpha polyubiquitination, and thereby it may impact its protein levels. Now we further confirm, because for an E3 ligase to target its separate substrate, it needs to interact with it. So we confirm that these two proteins interact with each other. So we perform this co-immunoprecipitation where we overexpress his CBP alpha, and in another condition we treated with MG132 to see if whatever CDK2 is present, if they are interacting with each other. So we found that in IgG there was no interaction, but in these two conditions there was interaction, and there was more interaction when MG132 was given. We further confirm that they also interact at endogenous levels. So CBP alpha, here we have used a cell line where we have inducibly expressed CBP alpha, and when you treat them with E2, the CBP alpha expression, the CBP alpha migrates to the nucleus. So we use that system, and here we showed that CBP alpha ER does interact with the CDK2. So far we confirm that CDK2 interacts with CBP alpha and apparently affects its protein levels through ubiquitination. Now, if CBP alpha expression is getting affected, does it also affect its function? To address that, we again went back to that in vitro system where we had used this K562 ER alpha inducible cell line. So this cell line is a cell line where we have CBP alpha tagged with the ligand binding domain of ER alpha, and this can be induced with the estradiol. So we use this cell system because what happens in this system, this CBP alpha ER is expressed but is present in the cytoplasm bound with the HSPs. The moment you give the E2 binds with the LBD region of CBP alpha ER size, and then it gets free from the HSPs and it migrates to the nucleus. And in the nucleus, it can bind to its target genes and it can induce the expression of those genes. So in this system also we confirmed that when we overexpress CDK2, this degradation of this protein was very faster. But in this condition, when we gave E2 here, these cells were differentiating. I mean, it is not clear here, but without E2, most of the cells were plastic in nature. In this, they differentiated and they took the neutrophil-like phenotype. And when we overexpress CDK2, again, most of the cells were blast-like. The image is not clear here, otherwise it's quite obvious here. So, taken together, so far our finding shows that CDK2 can inhibit CBP alpha expression, protein turnover, and thereby can impact its functions. Now we were intrigued to find out how it is doing it, how it is inhibiting CBP alpha protein level when it is not an E3 ligase. So while we were, contemplating about it, we came across an article that was published in EMBO in 2008 where they showed that CDK2 phosphorylates and stabilizes SKP2. Well, SKP2 is an E3 ligase and it is a key E3 ligase of the cell cycle pathway. So we thought that maybe CDK2 is stabilizing SKP2, and in turn SKP2 is targeting CBP alpha. Well, here I would also like to mention that in our previous study, we have shown that SKP2 is an E3 ligase for CBP alpha. So that we already knew that SKP2 does target CBP alpha. So we took that into account and then we first tested, is it so or not? So here what we found that when we overexpressed CBP alpha alone together with SKP2, CBP alpha was very nicely downregulated. And in this condition, when we treated with the roscobitin, even in the presence of SKP2, there was no inhibition, rather it was more stabilized. We also used some other inhibitors because this PD for P38 and SB for GSK3, because these are the two other kinases that were reported to phosphorylate CBP alpha. So here we observed that when we inhibit CDK2, SKP2 was unable to downregulate CBP alpha. So it gave us a clue that CDK2 might be downregulating CBP alpha by stabilizing SKP2, which in turn is downregulating CBP alpha. To further confirm if it is that way, we also depleted, first we overexpressed, because this was pharmacological inhibition, here we depleted. So what we did is CBP alpha alone, together with CDK2, there was downregulation of CBP alpha, and in this condition, when we depleted SKP2, CBP alpha was not downregulated, indicating that CDK2 indeed seems to be regulating CBP alpha via SKP2. Now, if it is so, we again used two different inhibitors. This time we used NU614, it is CDK2 specific, and then again roscovitin. So we used two different inhibitors to really confirm if it is happening that way. And what we found when we inhibited CDK2, SKP2 expression was downregulated. Even in this condition also, with roscovitin it was downregulated, but CBP alpha was slightly restored. We used two different inhibitors and we confirmed that SKP2 is indeed getting downregulated once we are inhibiting the CDK2. Now, to further show that CDK2 is indeed doing that way via regulating SKP2, here we overexpressed CBP alpha alone together with CDK2. And as you can see, with CDK2, there was phosphorylation of SKP2 at serine 64, which was already shown in that EMBO paper. So we just followed that paper. There was no change in overall SKP2 here. CDK2 was there because we had overexpressed, but CBP alpha was down. So it does confirm that CDK2 does regulate SKP2 and thereby CBP alpha. To further confirm if it is via the phosphorylation of SKP2, we also used two different mutants of SKP2, where we mutated all the possible phosphorylation sites that were phosphorylated by CDK2. And we found, particularly in this section, if you look, when we constitutively activated this SKP2, CBP alpha degradation was very faster. So this confirms that CDK2 stabilizes SKP2 by phosphorylating and might down-regulate it. We further confirm, it is not clear here, we further confirm that CBP alpha, CDK2 and SKP2 are present in the same immune complex through co-IP. While it is not clear here, you can see the bands are there in this condition. I'm really sorry for this because it is not clear here. Then we further confirm that CDK2 mediated downregulation of CBP alpha was indeed via ubiquitination through SKP2. If you look at these two lanes, this and this, here there is very heavy ubiquitination where there is only CDK2, while in this condition the SKP2 is depleted, so you don't see any ubiquitination in this. While rest everything is present in both the conditions, which shows that if you remove SKP2 and if you give CDK2, ubiquitination observed in case of CBP alpha is completely abolished. So this further confirms that CDK2 by stabilizing SKP2 through phosphorylation at serine 64 is stabilizing it. Once that is stabilized, that is ubiquitinating CBP alpha and degrading. We further confirm that when CBP alpha is down-regulated via CDK2 SKP2 axis, it is also impacting its transactivation potential through luciferases on a minimal PTK alpha loop. So you can see here with the SKP2, there was CBP alpha function was down-regulated, and with CDK2, it again down-regulated. In the last condition, when we gave CDK2 but depleted SKP2, the function was restored. We further confirmed through gel shift, where we showed that CBP alpha binding was there when CBP alpha alone was given, but with CDK2, because there was very less CBP alpha since it was down-regulated, there was no binding in this condition. This is cold flow, and in this condition, when we depleted SKP2, binding was again restored here. So this all confirms that CDK2 stabilizes SKP2, which in turn ubiquitinates CBP alpha and thereby impacts its protein turnover, which is reflected in its inhibited functions, which includes transactivation potential as well as differentiation inducing potential. Now, if it is so important, what will happen if we target both, SKP2 and CDK2? So what we observed that when we target both alone, the differentiation induction potential of U937 was just 2.4 to 18, but it almost synergized when we inhibited both together, SKP2 and CDK2. Which here, the differentiation CD11B increased up to 27%, indicating that targeting both CDK2 and SKP2 may lead to the robust expression of CBP alpha, which in turn may drive differentiation. This all findings we published in Journal of Cellular Biochemistry in 2020, and in the same year, another group published that CDK2 suppression synergizes with ATRA to overcome myeloid differentiation blockade in AML cells. In the same year, same month, they also published. So these two studies were going simultaneously. So it's like what we showed, they are corroborating those findings. So with this, I would like to stop here. Before that, I would like to thank my students Gatha Thakkar, who is here. This work was done by her, and for funding to the Lady Tata Memorial Trust and CSIR New Delhi. Thank you very much. I am open to questions. Well, I'm really sorry because slides were not easily visible there. Even I was finding tough to really correlate with this and whatever I was going to tell looking at those slides. So the idea was, please.
Any of the three molecules is any of them are being regulated in terms of level in AML?
Well, particularly with CBP alpha, there are two isoforms that are reported. Even in healthy persons, there are two isoforms, but the level of wild type CBP alpha, the bigger isoform is always higher compared to the another isoform, which is shorter isoform. But it has been observed that in leukemia, due to end region mutation, the shorter isoform is increased. So if there is changed ratio because of higher production of shorter isoform, it causes leukemia because it does not, because it still retains the DNA binding domain. So it binds to the all target genes where otherwise CBP alpha should bind, but because it lacks the N-terminal region, it cannot do the functions of growth arrest. So it acts as a dominant negative. So mutations are reported.
So you said that the CBP
Yeah. Well, SKP2 mutations are not known.
No, so binding site for the SKP.
In binding site for SKP2 in CBP alpha.
Yes.
Binding site for CBP, so because we have not done the domain mapping, so we have not yet shown to which region SKP2 binds to CBP alpha. So we are not aware of that.
Thank you, sir.
The therapeutic aspect could be that because the APL, which is acute promyelocytic leukemia, that is nowadays curable with differentiation therapy, but that is very small population. The leukemia, the AML M2 subtype, which is tough to treat because ATRA doesn't work there. So the idea is that can we sensitize those AML M2 subtypes with ATRA along with inhibiting either of these pathways. The goal of the game is like the AML M2 subtypes do not respond to the differentiation therapy. How can we sensitize them that they also respond? Because I mean 10 years back or few years back it was like uncurable APA, but now that is curable up to some cases. So this is what people are trying. If the differentiation arrest can be resolved, then most of the things can be resolved, particularly in case of AML M2 because they are really tough to treat.
For that, you will need cocktail of the inhibitors or single inhibitor will work.
I mean, along with the chemotherapy or differentiation therapy, either obviously cocktail will be needed because we are saying that CDK2 as well as SKP2 are target. So inhibitors again, these two, along with the ATRA or other different like IDH2 inhibitors can be given. So it has to be cocktail.
Then it may increase the cost of therapeutics. It may increase cost, so it will not be cost effective.
I mean, particularly in case of leukemia, where the window, for the treatment is so less, cost sometimes does not come because, well, it will not be that much costly like with the monoclonal antibody-based therapy, but because in AML, clinicians don't get that much time. It progresses so fast in the blast stage that patient dies. So even if you can extend that with a month or few months, that is desirable. From student side, either सबको समझ में आ गया तो किसी को समझ में नहीं आया. Well, if there are no more questions, I think I should stop here. Thank you very much.
Thank you, sir. It was such an intriguing lecture. Now, I would invite our next keynote speaker for the session, Dr. Santosh Kumar Mishra. Dr. Santosh Kumar Mishra is an assistant professor at the Department of Biological Sciences and Bioengineering, IIT Kanpur. He did his PhD at the Indian Institute of Science, Bangalore, and postdoctoral and research scientist training at the University of Illinois at Urbana-Champaign, USA. He is recipient of the prestigious Ramalingaswamy Fellowship from the Department of Biotechnology, Government of India, Professor T.R. Vishwanathan Young Faculty Fellowship in the Teaching Excellence from the IIT Kanpur and Research Project Findings from India Alliance, DBT, SCRBDST, DRDO, and other CSIR funders. He has more than 80 publications and many patents and book chapters to his credit. His research interests are biomaterials and biomedical devices. He will be today delivering talk on carbon materials as new hope for bio-nanomedicine.
Hello everyone, I am Santosh from IIT Kanpur. I know that I am the holding point between you and your lunch, so I'll try to be really quick. I'll try to share some of my work and some of the work which other researchers have done in the domain of carbon-based nanosystems. Before I start, I would like to thank the organizers for giving me this opportunity to interact such a knowledgeable audience. A special thanks to Professor Chaturvedi. So now, without further ado, today's topic, which I chose, it's kind of introducing a new kind of material, which is finally going to be decided by people like you that whether it qualifies to be a material for biological interest or not. We as researchers in different technology institutes, different scientific research institutes, we have our own hypotheses, on our ideas. We try to develop different things. Many a times it's the fascination of that material which pulls us in a particular direction. But the validation comes from the people like you who actually take it to the next level, who take it to the site where it is going to be used. And if we get suggestion, criticism, comments on those presentations, then we feel like, okay, now we are engaging with the right kind of people. So I would be expecting all this from this August audience. So let's start. I am associated with 3 different centers. I am faculty at Biological Sciences Bioengineering Department. Along with it, there are 2 more centers which have recently come up in IIT Kanpur. One is Gangawali School of Medical Sciences and Technology, which has the vision of bridging this gap between the medical centers and research labs. Another is Mehta Family Center for Engineering and Medicine, which is, you can say, extension of our bioengineering point. These are the people who actually work with us. So our current team has multiple PhD scholars, postdocs, RA, MTechs, and even BTech researchers who are doing their degree, and they do contribute in direction of research. Our collaborators, very important people who actually control us when we are running behind something without any particular aim or without any particular control, who tell us that, okay, no, this is correct or this is not. Because without being multidisciplinary, you cannot be successful in any of the fields. Then our funders, very important agencies who support our ideas. Sometimes these are very crazy ideas. And if you don't believe me, ask to my PhD scholars. So why carbon-based system? So if we just think about the carbon, then first thing comes to our mind that, okay, if we start from our house, if we go for our old cooking style of chulha, then there is production of this carbon soot. Then there is kajal, surma, different things. These all are carbon materials. Then we come out to go to our office or our labs, and then we are experiences the production of this carbon from automobile, a lot of carbon from factories and all. Then it just simply comes to our mind that, okay, it's something very negative. It cannot be something which can be utilized in biological system for some benefit. Now, this material has a lot of potential, but struggling with this kind of, you can say, given signature that, oh, this is something bad for biological system. Now, some of the researchers in last decade, in the beginning of last decade, took it as a challenge, and they thought, okay, let's check when the whole body is made up of carbon as atom or element, and it is just threading with carbon, with hydrogen, oxygen, phosphorus, sulfur, and creating different molecules. How can carbon itself be that bad? There must be something in those carbon materials which could be used for the benefit of biological systems and ultimate benefit of the human being, because we are the one of most greedy, most species in this world. We always think about what is good for the human being. So that is the criteria that, okay, whether it is good for human system or not. Then they started coming up with different kind of molecules which are based on carbon, and carbon itself was found in multiple allotropic forms. So it was either carbon nanotube, it was either graphene, it was buckyballs, and all these materials. The biggest challenge these carbon material scientists they faced, that it was not soluble in water. And anything which is not soluble in water, either you have to assist the solubilization by using something which is organic, and that even if it has excipient, it will create toxicity, or you change the carbon material so that it becomes soluble in water. Then different strategies came, and they started generating a new form of carbon material, which is called carbon nanoparticles. Now, these carbon nanoparticles, luckily, they are suspendable in water system. So you can suspend it in regular water, you can suspend in buffers of different pH, you can suspend in different growth mediums and different body fluids. Now, that was the barrier which broke the restriction of carbon material in non-biological system use, and it came to biological system. Now, today I'm going to talk about few of the properties of these carbon nanoparticles and see whether it is qualifying to be a new hope for the biological bio-nanomedicine for use in clinics. Here there are some examples. quoted here. So they can be, this is just the highlight so that we can get more engaged in this material. So it can be used for bioimaging, it can be used for biosensing, it can be used for drug delivery, it can be used for making smart polymeric materials, gels, transdermal therapeutic, and all that. Now, if it is so promising, how easy or difficult is the preparation of this material? Because that will be the first bottleneck which one want to handle. Because if it is very difficult to produce, then its cost is going to be higher, and then it will be less available, more skill set would be required to produce it, and so on. Luckily, it is very easy to produce. If you have a hot plate in your lab, you can produce it. Forget about hot plate. If you have a kitchen microwave in your lab, you can produce it. Even forget about that. If I say that every day, if you are baking something in your kitchen, you are actually producing these carbon nanoparticles. So I'm not saying wrong. Because whenever you burn any sugar or any carbohydrate or any protein or any fat, ultimately you are producing carbon nanoparticles. Now the only trick is that how to control the synthesis, make it reproducible and isolate it, because there will be a lot of chunk of carbon which will be produced, and that you cannot use as in any of these applications, for any of these applications. Now, if you want to really make it more sophisticated, more controlled, then there are microwave synthesizers which can be used. So generally in our lab condition, we use it. But sometimes for experimental purposes, for scaling up the process, we also use the hot plate method or kitchen microwave method, so that we can see how easily it can be produced. Now there could be a question that, okay, if you are using very purified carbohydrate material, it could be very costly to produce. So there is alternative. There are multiple byproducts of different sugar industries and carbohydrate industries and protein industries, food industries, which can be used as raw material to produce it, like molasses, like syrup, like nectar, like honey, like peel of banana, even peel of different fruits. They can be used to generate these carbon materials. Only thing is yield percentage, quality of particle, size, their surface chemistry that might vary, that you have to tune according to your requirement. Now, if they have to qualify to be good for biological system, they need to interact with cellular system. Now, do they interact? So answer is yes, they do interact. They enter cellular compartment and generally they take endocytotic pathway. So that has been proven in one of our studies. We could prove it. We use different kind of blockers of different entry routes. And we saw that, okay, which are the particular pathways it is following. Now, it depended on that what kind of surface chemistry you are generating during the preparation. So that tells that there is a tunability. So it's not that whatever the original material you are using to produce them, it's also that after producing you can coat with different kind of materials and then tune the property. And that will be entering in the cell by different pathways. Then you want to know that how you can use it for bioimaging application. So these particles, these carbon materials, these carbon nanoparticles, they are autofluorescent. So what they do, if you excite them in the range of 350 to 550, they start emitting light in just plus 50 nanometer of wavelength. So you start 350, you get light at 400. You excite 400, you get light at 450. You excite 550, you get light at 600. Now that is the limit where one need to push it forward, because we know that for our biological tissue, our blood, the maximum absorption of light in that range 500 to 600. So you want to push it beyond. For that, you have to incorporate or passivate different kind of molecules, polymers, dye molecules, pigments, so that it can be shifted toward near IR. And then it becomes a very magical bioimaging agent, which can be also used for 2-photon imaging, which can be used for deep tissue penetrated system and imaging. Now, how would you know and how would you utilize it in your cellular system? So there is an on-off system can be done, because many of the physicists, they did multiple research and found out that whatever fluorescence or luminescence is coming out of this system is surface phenomenon. And if you decorate the surface with any molecule which can quench those catars created on the surface, then it will be off. And then as soon it goes away, those molecules are going away, it switches on. So if you want to follow any biological phenomenon inside the cell, where there is a particular molecule which can make association with your carbon nanoparticles, then it can work as a switch on and switch off based on the which stage of that biological phenomenon you are in. Okay, so that's how it can work. We could investigate it and publish it. Then how to change the surface passivation? It's very easy. You can do during the preparation of carbon particles or after the preparation of particles. So suppose you are interested in a large molecule or large protein or large gene sequence which you want to send inside the biological system. You prepare these particles and then decorate those polymeric materials, could be biopolymer or synthetic polymer, and then that wrapped system, you send it inside the cell. Now when it will reach to endosomal cavity, there because of the presence of these anionic lipids, it starts interaction, and then because of ion pairing, during the ion pairing process, it kicks out those molecules which are wrapped around. Now, if it is a protein system, then you do that kind, you use it for that particular therapy process. If it is gene sequence, then it can go either you might be delivering siRNA, shRNA, or you want to deliver the DNA, which can go inside the nucleus and start the process. So it is very easy. Now, if you are doing co-preparation, you can even tune the size. The size could vary from 2 nanometer to 200 nanometer. Now that also is very reproducible and yield is also very nice. So it depends on whether you want to use it for just biofluid purposes or you want to deliver some content inside the cell. You can decide, and based on the pre-decided composition, you can get a size. Right? So in generally nanomedicine, the concept is if you want to deliver something inside the cell, then the size should be close to 100 or less than 100, because the leaky vasculature, like in case of the cancer, right? If you want to deliver a drug loaded on these nanoparticles, it has to cross that leaky vasculature, it should be less than 100. But in some other cases, even the larger one would work when you do not, where you don't want to go inside the cell, right? So size variation can be done. Now, what about if you want to use intrinsic property of some medicinal plants? And you don't want to extract that medicine or that active component from the plant, but still you want it to be incorporated in the nanosystem. You use that raw plant part. It could be leaf, it could be crust stem, it could be roots, it could be any part of the plant or a plant as such, and then convert it to carbon nanoparticle. You'll find that many of those particles, they will inculcate that medicinal property. Now, there could be a question that what about the heat you are generating or the microwave you are exposing it to, that might degrade the medicinal value of that particular active component. So this is a very genuine concern. So the option is that you do microwave preparation. Many of these thermolabile materials, they are stable in microwave. So that increases the bandwidth of this process. And then more and more materials and more active compounds can survive. So here we used an algae for preparing this carbon particle, and these carbon particles were so good by themselves, they had the property of controlling the migration of the cells. So if there is a chance of metastasis, then if this particle itself is used directly without adding any drug, it works as anti-migratory. Now, yeah, how they do interact with cellular membrane? Now, by themselves, they will not stay in the membrane for a long time. But if you want it to stay there, you just conjugate a long chain fatty acid on the surface. And then because of the fatty acid chain, hydrocarbon chain, interacting with the hydrocarbon part of the membrane, it can stay there for longer time, right? So that stay period you can increase. How can you utilize it for intracellular interactions? So nowadays, a special technology has turned up for the nanomedicine that is called nano cases of DNA, right? So DNA origami. So DNA-based nanotechnology. Now, what happens? You have a particular sequence on that nanosystem. That same nanosystem can be created on these nanoparticles because they have autofluorescence, and their fluorescence you can tune that from the same batch of particles. One particular population is giving green fluorescence, another is red fluorescence. You tag them with different sequences when they are inside the cell, and if they are interacting, there will be fluorescence quenching, and you can follow it, you can quantify it, that how much interaction between those two long chain nucleotides is happening inside the cell. You can, another concern, which is very genuine concern, that what happens, whether it is safe or not, what happens, what is the fate of these particles. So we started investigating that what happens to these carbon materials where they are incubated with biofluids when there are some particular enzymes available, and we found that they are completely degradable. They degrade inside the body and they degrade in soil because the peroxidase enzymes, they can chew them off. And we did follow the pattern of Raman spectrum to find out what is happening to the integrity of the carbon structure. We found that it was completely chewed off within 7 days in biological system or cellular system, you can say, and in soil, it horseradish peroxidase, which completely cleaves it off in 24 days. So basically, it is a sustainable technology which doesn't leave its own carbon footprint, because nowadays it is one of the catches, what's your carbon footprint, right? So this carbon material doesn't leave its own carbon footprint after being used or after being discarded. Then how can you quantify that how much drug is being actually used in the cellular system? So most of the side effects which we get out of medication is because of the overdosing, because we do not know. And the process of delivering is also, it's so unpredictable that once you inject, by the time it reaches to that particular tissue, only 4 to 5% is being used. Rest 94% or close to 95% is being cleared off from the system. Now that how to avoid overdosing and finding out what is the amount of drug in that particular state exactly required to do that either protein activation or deactivation. So these, because these nanoparticles, they have their chemical signature. So there are some spectroscopic methods which can be used for screening those drugs in a particular cell type in laboratories, and then can be given to doctor, and then doctor can give suggestion based on that, that okay, doses should be increased or decreased. So there is, here we used hyperspectral imaging, and we could read the chemical signature of these nanoparticles and the drug which was loaded on it, and we could see the separation time, that how much time it takes to separate and what amount of drug actually got separated, and based on that we could decide our doses, and we did the animal experiment, and which was more successful rather than when we were doing without estimation. Then as I said, it can be used for 2-photon bioimaging, just the surface passivation of polylysine, and the carbon materials are having very long half-life time when you are exposing it under a microscope for doing 2-photon. So that was really nice. Then it can be also used as not directly as a therapy, but as a supplement. Now, what happens? One of the biggest problem is the drug resistance, and that drug resistance is not only problem of the eukaryotic cells, even prokaryotic cells. So, antimicrobial resistance is one of the domains. So, what this material can do? It can carry different kind of salt content, different kind of ions, and make cells more susceptible. And then you can do the secondary therapy, whichever you have decided, and it will be more working. So here we try to do it for the GSX modulation, and we could deliver iron content, iron ions, entrapped on these nanoparticles using different kind of polyphenols. And we got the success in that, and we could improve the therapeutic potential of some of the conventional drugs. Then some gel material. Now we know that for the muscles, if there is a mechanical injury, if you leave it for a few weeks, by itself it will be cured. But right now, the way our country and all across the world, people are involved in sports and athletics, even one particular injury for one particular match can cost somebody a gold medal or somebody a very good big trophy. They don't want their player to wait for that long to recover. Now for that, there are some gel materials which can be used. Now, till now, what is the process? That you give some medicine, but the problem is that sometimes because of the ignorance, those drug molecules can create problem in the dope test. And without knowing, one of the metabolite of that drug can be caught in the doping scandal, and then that person is banned. Now that compels people to generate non-drug based material for regeneration of muscles. So we generated penipam based or nipam based polymeric system gel, which could be used for enhancing the differentiation of myoblast to myotube. But the problem is that the strength of the material and the process speeding is not that much. So what we did, we used carbon material, and this carbon nanoparticle, when it went inside the gel, it not only improved the mechanical strength, but the adhesion and the differentiation process got speed. And within 72 hours, we could recover some muscle injuries, which generally doesn't get cured up to 10 days or so. We did DOMS model of animal, and there we showed all the biological parameters, including the biochemistries and histopathology. And we did extensive mechanical testing by using atomic force microscopy and other methods. Then these carbon materials can be included in biodegradable drug-eluting cardiac stents, where it can provide the strength, it can work as a reservoir for delivering the drug, and whenever there is degradation of the content, slowly it can release the carbon particle, and those carbon particles are further getting degraded and will be in the system for longer time. So it can be used there as well. So these are the ways we did it. Different kind of, I know that I'm a little late in that, but we did all kind of characterization. We got successful. We finally used it in ex vivo condition of the pig heart, and we could use those structures which we have created by 3D printing. These are some of the other technologies which are working, and these carbon nanomaterials are really handy in those cases as well. We are making medipatches, we are making medicaps. Those can be used for personalized medicine. That is another catchy word, but everything boils down to a very nice, sophisticated material which can do the job. We also generate biosensing platforms where we use this carbon material to coat on filter paper, and that paper converts to a biosensing platform. So that brings down the cost, and that also becomes like use and throw, and you can put multiple detecting molecules using antibodies or aptamers or peptide sequences to recognize a particular biomolecule which could be present in any of these biofluids, or including urine, sweat, saliva, or tear, or blood, and then you can find out. So these are some of the examples and patented technologies which we produced. So finally to conclusion and way forward. So you need a therapeutic regime, you need a method, but you also need a material composite which can do job for you. And thank you very much for your kind attention. And I'm open for questions and comments and suggestions, please.
Thank you, Dr. Sandesh. I have one very basic question that you mentioned that it is biodegradable, which I am not sure it's true or not. So, what is the fate of these carbon nanoparticles in biological system? What about the bioaccumulation, and whether they can cross the blood-brain barrier, and how they will act in fine micro vessels, particularly brain micro vessels?
Thank you. So it's very loaded question and so many questions in one. But yes, I will try to answer one by one. So being any material biodegradable, the way we assess it, we started with our in vitro system. So we first created simulated fluids of different type, different enzyme composition. We impregnated it with different salt content and all, and we found that the chemical signature which comes from the carbon material that gives very specific G and D bands in Raman structure. So when we followed it with the timeline, then we found that slowly those peaks were disappearing, and at the end of the 7 days, that completely disappeared. Second, that's how we thought that okay, it can be used in the biology system. Then we did the study in animal model, where we loaded some of the cancer drugs on these nanoparticles and we tried to follow what is happening. Now, what did we see? That because whenever they are not tagged with any targeting moiety, then they do go to some of the other organs, like some in liver, some in heart, some in intestine, some in spleen. We could collect, we could see those very, I would say, fragile features. But when we had targeting, so what did we do? We used some peptide sequences which were meant for targeting a particular xenograft tumor which were developed in the animal model. And we could see that more than 90% content was going straight to the system, to that location. So it was the targeting moiety which was deciding where it will get accumulated. Even if it is getting accumulated, whether it has side effect or not, that also we check. So in every tissue we followed with the timeline, and we found that the content was decreasing, the chemical feature. Again, as you said, that until unless it goes to really in the hands of the people who do work in the clinic, I do not know. But in my assessment, that it was degrading in all those tissues systems where it was going. Now, third question is whether it can cross blood-brain barrier. Now, for crossing blood-brain barrier, there are two parameters. One, the lipophilicity of the system, and another is the size. Now, if you do not want it to cross, make a size which doesn't allow it to cross there, or the lipophilicity of the system also can be tuned. Now, it entirely based on your design. You want it to cross, it will cross. You don't want it to cross, it doesn't cross. Because this particle is not that soft as in case of liposomes, lipid-based or polymer-based. It is a semi-soft, I would say. So it is somewhere in between the metal nanoparticles and soft nanoparticles. So now it is the squeezing property of this particle is not that much as case of the polymers and all. Though they are bigger in size, they are found to cross the blood-brain barrier and cause the toxicity. But this system is like if you, so the best example is when we did hydrodynamic diameter in the suspension system. Suppose we are getting 80 nanometer as size. And even if we do TM, it comes very close to 75. So the water of hydration is not too much in the system. So it remains as such. So now you can tune the size which you want it to cross or you do not want it to cross. Surface chemistry you can change based on lipophilicity or hydrophilicity. So this is now what will happen if it goes to the final categories. Now there is a concept of corona formation. So what happens when these particles, any nanoparticle comes in contact with the blood or any other biofluid, what they do, they start accumulating protein molecules on the surface. We did that study and we found that whenever it is positive charge, corona is really humongous. It's like thick. When it is negatively charged, like you have decorated phosphate, you have sulfate, then they don't make corona, right? So if you want to deliver it in the finer capillaries without creating the corona and the coagulation, then you should have a carbon particle which is negatively charged, and that might be beneficial. But we have not explored it in those kind of real conditions, so we cannot say. But this is my presumption that this might be the case. I hope I could answer some of the questions. Thank you.
Regarding the blood-brain barrier, you
Right.
said the size. Actually, blood-brain barrier have types.
Yeah, exactly.
Size doesn't matter there. There are two things: lack of visibility, perceptivism, or active person. So, blood-brain barrier, these two mechanisms are.
Absolutely, thanks. The size I said because see, even by diffusion, so when you go to a small
Lack of visibility.
Right, because these particles can be even dot form, so they are called carbon dots. They'll be like 1 nanometer. 1 nanometer is 10 angstrom. So even if it is tight junction, there, because there is possibility of diffusion, they might diffuse rather than going endocytosis. No, that's what. No, if you make that size, but if you go beyond 50, then it will not go there. So you have to tune the size in that. But thanks for your comment. Okay, so I think no more questions, and thank you very much for patiently listening to me. And it is great delivering a lecture in this August place and between August audience. Thank you very much.
Thank you, sir. It was indeed an interesting lecture. Due to time constraint, we won't be having an online lecture. Now, I request Dr. C.P. Chaturvedi to felicitate Dr. Arun Kumar Trivedi. I again request Dr. Chaturvedi to felicitate Dr. Santosh Kumar Mishra. Thank you, sir. Now I would request Dr. Chaturvedi, faculty in charge research, to express his vote of thanks.
So very good afternoon to everybody, and it was indeed like a very great pleasure that this research day has been celebrated in a very good way, and we have a lot of audience, lot of participation, and it makes us happy because our team has been working hard to make this event like good since almost like 10 days. So it's time for to close this session. Before that, I would like to basically acknowledge all those people who have been behind this conducting this session. Especially, I would like to start with my director and dean who have been supporting, whose vision has been there to start this this event since almost last 3-4 years back, and it is growing year by year, and it is very happy that every year the participation is going high. And then I would like to thank all the judges from the various institutions who have been, actually it has been really pleasure that they have come here, they have came, devoted time and listened to the work of the students as well as the faculty which is being carried out at this premier institution, and for spending there and sparing their time. I would like to thank them all. I would like to thank all the students and the faculty member of the institution, without participation of whom this event had not been successful, because the participation of any event make it basically successful. So great thanks to all the faculty members and the students. I would like to thank all the people, all the committee members of this research organizing committee, Dr. Jalaj, Dr. Santosh, and all research cell staff, ethics cell staff who have been really doing hard work to carry out this event. I would like to thank the electronic media. They were covering it outside. And I would like to also thank, most importantly, our two speakers, Dr. Trivedi and Dr. Mishra. Dr. Mishra, who came from IIT Kanpur over here, and he has delivered such a nice talk on nanoparticles and their application in biomedicine. It was a great pleasure to hear his talk. Same Dr. Arun Trivedi, he is from the CDRI. So I would like to thank them also for coming over here, sparing the time and letting the audience with the new insights into the different area of science. And finally, I would like to thank you everyone for this event, and the event is now closed and you all are invited for the lunch. Thank you so much.